Hi, welcome back. I'm here to talk more about PureTarget in action: accurate, powerful, and efficient genotyping. We've learned about PacBio HiFi sequencing and the PureTarget enrichment method. Now, let's see how PureTarget performs in real-world applications. We'll focus on its scalability, accuracy, power, and efficiency, especially for challenging repeat expansion genotyping. One major advantage of PureTarget is its scalability. Consider screening for ataxia, which can be caused by expansions in over a dozen different genes. Traditionally, labs might run multiple single-gene tests, like repeat primed PCR or Southern blots. This is often laborious and time-consuming. The PureTarget repeat expansion panel, however, covers 20 relevant genes, including many ataxia targets, in a single reaction. You can multiplex up to 48 samples per sequencing run on a Revio or Vega system. This consolidation dramatically streamlines the workflow, replacing potentially dozens of individual tests with one modern, powerful long-read assay. This scalability and efficiency is being recognized. Major diagnostic labs like Athena Diagnostics, a Quest Diagnostics business, recently announced that they are using PureTarget technology as part of their upgraded ataxia offering. They explicitly cited the streamlined workflow and the ability to process larger sample pools as key benefits, alongside improved analytical capabilities like methylation analysis. But is it accurate and consistent? We benchmarked PureTarget using well-characterized reference samples from the Coriell Institute, which contain known repeat expansions. We prepared libraries and sequenced them multiple times across different PacBio platforms: Revio, Vega, and Sequel IIe. The results showed excellent consistency. For the shorter, non-expanded alleles, we saw 100% concordance in repeat counts across all platforms and replicates. For the longer, expanded alleles, the counts were consistently within 10% across platforms. This is well within the range needed for accurate, pathogenic classification, with any minor variations often attributable to known biological mosaicism in the samples. Sometimes, HiFi sequencing provides more detail than older methods. Take this ATN1 example. The expected genotype based on classical methods was 16 and 68 CAG repeats. PureTarget consistently called 20 and 72 repeats. Why the difference? Looking closely at the HiFi sequencing data using the target plot, we clearly see the expected CAG repeats. But we also see two CAA interruption motifs near the beginning of the repeat tract on both alleles. It turns out that the classical assay only counted the pure CAG run, omitting the first four motifs: CAG, CAA, CAG, CAA. When we account for this difference in definition, the PureTarget results perfectly match the classical count. This highlights that repeat definitions matter. And while some recalibration might be needed when adopting a new repeat expansion genotyping technology, HiFi provides the full sequence resolution to explain any discrepancies. PureTarget also demonstrates immense power in resolving highly complex repeats. RFC1, associated with Canvas, is notoriously difficult. It features very long expansions, potentially over 15 kb, extreme motif diversity, multiple benign and pathogenic motifs, and GA-rich sequences prone to secondary structures. PureTarget, being PCR-free and long-read, can sequence through these challenging regions. We've successfully sequenced double pathogenic expansions over 6 kb long. In one fascinating case provided by collaborators, RP-PCR suggested a pathogenic expansion. However, PureTarget revealed that the expansion consisted of a different, likely non-pathogenic motif that the PCR assay couldn't distinguish. This level of resolution is critical for accurate interpretation. Finally, let's talk efficiency, using FMR1 associated with Fragile X syndrome as an example. Accurate FMR1 genotyping requires knowing three things. First, the repeat size, which can be very long and GC-rich. Second, the presence of stabilizing AGG interruption sequences, which are important for predicting expansion risk. And third, the methylation status of the promoter, which is linked to gene silencing. Traditionally, labs might need separate assays, perhaps PCR or southern blotting for sizing, another PCR for interruptions, and a methylation-specific assay. With PureTarget, because it uses long reads on native DNA, you get all three pieces of information from a single multiplexable test. You accurately size the repeat, you directly sequence the interruptions, and you get the methylation status, a true 3-in-1 assay. And again, the results are highly consistent, as shown here with multiple technical replicates for FMR1 sizing across different instruments. So to summarize, PureTarget coupled with HiFi sequencing is proving itself in demanding applications. It's scalable, enabling streamlined workflows adopted by major labs. It's accurate and reproducible across platforms. It's powerful enough to resolve extreme complex repeats and sequence features missed by older methods. And it's efficient, consolidating multiple assays into one comprehensive analysis.